I think we can get started. So welcome everyone. Thank you for joining us on our Variant Factors seminar series. I am Adriane de Souza. I am a member of the organizing committee, along with Ian, Diego, Mariano, and Ohana. And these seminars are made possible with the help of Lara Muffley and Alex Hopkin, and also with all the support of our sponsor, the Atlas of Variant Effect Alliance. As a reminder, we are always looking for potential speakers. If there's someone you like to see presenting, or if you want to share your research and your work, feel free to submit a nomination in the speaker nomination form. There's a QR code here, and we'll post the link in the chat as well. But before we start, there is some information I want to share with you. Each talk is around 20 to 25 minutes. There will be time for questions, and please feel free to post your questions at any time through the Zoom Q&A, and we'll read your question to the speaker at the end of each presentation. If you have other questions after the end of the presentation, you want to talk about it, you can send them in our Slack. Here is the Slack channel for the Atlas of Variant Effect Alliance. And you can also follow us on Twitter to get updates. We are always posting reminders about the Variant Effect Seminar Series and all details of the presentation. Additionally, we post all the full presentations on YouTube, and here is a QR code so you can follow the link to our YouTube channel, so you can find past presentations as well. And lastly, as a reminder, we have the VESS schedule available. Every month is updated in our website, so you can use this QR code to see the details and the description of our speakers that are aligned for the next months. And we wanted you to, as a reminder, be considerate when submitting comments or questions. We expect everyone to be respectful and use welcoming language and inclusive language. Now we can go ahead with our presentations. So today we have two amazing speakers. So the first one is Sriram Pendyala. He majored in chemistry and physics in Boston, and now he is at the Fowler Lab in Seattle, and he's developing technology to study protein variation using microscopy. And after Sriram, we will have Phil Huss, and he is a doctor and now a postdoctoral fellow in the Raman Lab in the University of Wisconsin-Madison. He studies bacteriophage host interactions in the context of genetic perturbations that can change sequencing to function landscape of these interactions. So without further ado, welcome, Sriram. And if you're ready, you can start sharing your screen.
All right. Can everybody see the title slide?
Yes, go ahead.
All right. So today I'm going to be talking about a project that's been going on for a few years, where we're trying to develop a method in the Fowler Lab to be able to do pooled optical phenotyping of variants. So meaning like you can take a library of variants in a protein, tag them to something like a GFP or a flag tag, and then image them in cells and look at how different variants might localize, say, to different parts of the cell, or maybe induce cellular changes that you can see with microscopy. So the project isn't done yet. I think we're about halfway through. So unfortunately, if you're looking forward to seeing the full heat map, we won't quite have that, although we're analyzing the data. So just fair warning. But I will talk some about the technical challenges that we've had to overcome in the project. So I'll start with an example of one of the diseases and proteins that we're looking at, which is a protein that causes dilated cardiomyopathy. So here is a normal looking heart at autopsy. And here's one from a patient who suffered from dilated cardiomyopathy. And so you can kind of see that both of the heart ventricles are dilated, and the thickness of the muscle that beats in the ventricles is low. And so that causes there to be heart failure, basically. And so it's a really relatively common genetic disease with a high incidence. And there's lots of variants of uncertain significance in these cardiomyopathy syndromes, both in hypertrophic and dilated cardiomyopathy. And so that's like one reason why we want to do mutational scans of proteins that cause these syndromes. And in addition, of course, with any sort of clinical genetic disease, one of the ways to be able to diagnose patients is to map the segregation or co-inheritance of an allele and the disease. However, we don't always have the sort of family data that we would need to do this, and that's what the whole VESS is about trying to phenotype variants, and this is one reason why it would be so amazing to be able to phenotype thousands of variants in proteins that cause diseases like this. So the protein I'm interested in is the nuclear laminar protein, Lamin A. And what's, I think, really cool about this protein, but also unique, is that germline mutations in this protein can cause numerous different diseases. Of course, dilated cardiomyopathy I just mentioned, but also muscular dystrophy, lipodystrophy, and this very rare disease called progeria. Have you ever seen pictures on the internet of 10 or 15-year-olds who look like they're 90 years old? That's due to this disease. And so I think that what makes this protein special is just the number of different diseases and different tissue types that it can cause, even though it's expressed in all differentiated cell types. And that makes it somewhat different than something like BRCA1, which is also expressed in a whole bunch of different tissue types and cell types, but really causes the hereditary breast and ovarian cancer syndromes. And so in that case, a bunch of people from the University of Washington did multiple mutational scans of different regions of that protein, looking at one phenotype. But here we kind of need to really think hard about exactly what phenotype we want to look at, because it's not entirely clear what even the mechanism of pathogenesis is for any of these diseases. So Lamin A encodes this structural protein. It's an intermediate filament, a type of intermediate filament that forms the inside of the nuclear membrane. So there's two leaflets for the nuclear membrane, and on the inside of in the inner membrane, in the inner leaflet of that membrane, this protein sits and forms like this structural fibrillar network. You can see right here, and it interacts with other sort of cellular cytoskeletal elements like actin, and which then interact in turn with sarcomeric elements. So it interacts with actin indirectly through a bunch of other proteins, but maybe this is one reason why it's causing these cardiomyopathy syndromes. So something that people noticed in the last 2 to 3 decades is that if you take fibroblasts from patients that have these Lamin A-caused dilated cardiomyopathy and stain for the expression of Lamin A as a protein, you see these sort of protein aggregates that appear on the nuclear membrane instead of what it should look like at the wild type version of the protein, where you kind of just get this coating that appears on the inside of the nucleus. So these pathogenic variants seem to undergo this aggregation in primary human fibroblasts. So this is kind of the optical phenotype that we wanted to go for. And so what we did is we expressed the same variants tagged to a fluorescent protein in U2S cells, and we also transfected those variants into iPS-derived cardiomyocytes. And when you do that with the wild-type lamin, you get this sort of wild-type morphology where it kind of sits exactly where you expect in the inner side of the nuclear lamina, or the membrane in the nuclear lamina. But if you express this N195K variant that causes cardiomyopathy, you get these aggregates which I thought we were really happy to see, that were recapitulating the phenotype, both in expressing in U2S and also transfecting into cardiomyocytes. And then when you express this muscular dystrophy variant, which we only express in U2S cells, you also get the aggregation phenotype. But when you look at the lipodystrophy and progeria variants, and specifically this is an atypical progeria variant, but we also looked at progerin, which is the main progeria causing variant, you don't seem to get this aggregation phenotype, although you may get other phenotypes. And so what was seemed like to us was that the variant aggregation appears to be specific to the myopathy causing variants, although we've only tested 2 here. So that both the muscular dystrophy and the cardiomyopathy. And then there was this recent paper that was published. I'm not going to talk too much about it, but they've tested a whole bunch more variants and found that the aggregation phenotype really does seem to predict, but not 100%, is not 100% in the pathogenic myopathy causing variants. So this is a pretty good phenotype to go after. And so now the question is, as I mentioned before, if we were just to try to do this one variant at a time in cardiomyocytes, this would take us, decades to try to transfect every single variant in one at a time. So what we would like to do is be able to do this in a pool for all of the 13,000 missense variants of the protein. So how do we do that? Well, the first thing we did is we created a library we bought variants. We actually just bought a library from Twist, cloned it in, and tagged it with this GFP, and then just looked at it in U2S cells. So once you do that, you get this sort of picture where you see variants that seem to have different amounts of aggregation, or excuse me, you see cells that seem to have different amounts of aggregation. So examples are cells that have more aggregates and cells that don't have aggregates. And so, of course, like what we would like to do, and we kind of came up with a few different ways to do this and tried actually some different ways to do it, is really be able to say somehow that the variance in this yellow circle, there's some that corresponds to some variant, and the variant in the blue circle corresponds to some other variant. And so how do we kind of get the genotype information back? You see the image and you kind of want to be able to get the genotype information back. And so around this time when we were playing around with some other methods, there was this paper that was published to do in situ sequencing to be able to recover genotype information from cells. So they were doing it in the context of these CRISPR-Cas9 screens, where they would introduce, say, one guide per cell and create a pooled library of cells, each expressing one guide. And then here's what they would do. And I'm going to explain the in situ sequencing workflow and show you some of the data next. So each cell expresses, say, a guide, and that guide is expressed from DNA into RNA, right? And they actually set it up so it's expressed both by a Pol3, which is actually creating the guide RNA, and a Pol2 promoter, which creates this whole long transcript, which has like GFP and a pyrrho resistance gene, and also the guide RNA. And what they're interested in is the Pol2 transcript. But suffice it to say that there's many RNAs in that cell that have that Pol2 transcript that has the guide RNA, which they want to capture. So in our case, it's actually a protein variant that we care about. So we have a GFP tagged Lamin A variant, and then there's a barcode that maps to that Lamin A variant that we can subassemble using PacBio that's expressed on the same RNA as that variant initially. And so there's many copies of this RNA in a cell. So they do in situ reverse transcription and convert this RNA to DNA. And then what they do is they align this thing called a padlock probe or a molecular inversion probe, a MIP, to this cDNA that you get. So this padlock probe, it has 5' homology. It's just a single-strand DNA, just like any other oligo. And it has some 5' homology to the 3' padlock sequence and 3' homology to the 5' padlock sequence present on the cDNA. So you get this sort of partially double-stranded, partially single-stranded circle that forms in situ. And then you can just add a polymerase and a ligase. And what'll happen is the polymerase will extend the padlock and add in the cognate complement of the barcode. And then the ligase will ligate the padlock shut. And then now you have this single strand circle that has your barcode sequence in it. And so once you have a single strand circle, you can do something called rolling circle amplification, where you copy the circle thousands of times using 529, which is a rolling circle polymerase, that the same one they use, an engineered version probably, in the PacBio sequencing. And so now you have this red single-strand DNA, long single-strand DNA kind of nanoball that contains thousands of copies of your barcode sequence. So now you've kind of done what they managed, similar to what they managed to do in an Illumina flow cell, where you've copied your barcode or whatever, your amplicon sequence thousands of times, so now you can sequence it. And they did sequencing by synthesis, just taking Illumina MySeq kits, which is exactly what we do, and just basically removing the reagents from them, the incorporation and cleavage reagents, and pipetting them directly onto either well plates or slides. And so this protocol was derived from the initial PhysSeq protocol that was published by the Church Lab in 2014. And they came up with a version to do these pooled optical screens by 2020. And so, of course, they were using this for guide screens, but we want to use it for protein variant screens. And so, of course, once you add your sequencing primer, you can then start doing in situ sequencing, and you can use the microscope to be able to do the sequencing of your sample. So here's what it looks like. Here's what a single base of the Illumina sequencing in a well plate looks like. You get these 4 colors in the sequencing, and there's dots that correspond to reads in every cell. So you can see these different cells here. And the color of the dot tells us which base we're reading in that read, just like an Illumina machine. But of course, we need to do this many times. So we do this for the entire sample, we incorporate an image base 1, then we cleave, and then we need to incorporate an image base 2, and then cleave that, et cetera. And we need to do that for as many bases as will tell us the identity of every variant in the library. In our cases, it's 12 bases. So this is quite an involved procedure. But once you start getting cranking, you can really do this in probably about a week, the entire protocol. Okay, so we first tried this, and we used our landing pad, which I'll talk about later in the talk, and just put our, and that's basically a genomic location where you can then use a recombinase to move specific, payloads and express them via Pol2 under the control of a promoter, 1 per cell. And so what we did initially was exactly what I showed you, which is putting the barcode on the linear RNA that also contains the protein variant. And unfortunately, what we saw was that we're getting a lot less reads per cell than the original Feldman and Singh cell paper, where they did this for guide RNAs. And so we kind of reasoned that it might be the case that our landing pad expression is just lower than the expression that they're getting off the viral promoter or off the CMV promoter, whichever promoter they use in these guide screens. And that just leads to a lot less reads per cell. So you can kind of see, say, base 1 here. And so you get a lot less read per cell and then a lot lower fraction of cells that you can map to the library. So this is kind of the first challenge that we had to go out and tackle. And so how do you boost the number of reads per cell you get? Well, we didn't really want to, say, massively boost the amount of the RNA expressed, because that would massively boost the expression of the transgene. It might change, actually, the aggregation status of the cells. So we wanted to be a little bit more careful than that, and be able to kind of decouple the RNA that we're capturing from the RNA that expresses the protein variant of interest. And so to do that, I saw this, actually, I saw this paper from JB and Sam in the Chanduri lab. It's actually coming out in Nature Methods this year, I believe. And they use this circular RNA tornado circularization technology. It uses tornado ribozymes to circularize a small RNA, which was expounded in this Nature Biotech article in 2019, where they have a U6 promoter that expresses an RNA that has some circularization region flanked by some ribozyme sequences and ligation sequences. So when you express this off of a Pol3 promoter, the eventual product is this circular RNA. And the nice thing about circular RNAs is that a lot of the RNases in cells that degrade RNAs degrade them exonucleolytically. So when you have a circular RNA, you can kind of build up to high levels in the cell. And one special advantage of their technology is that from this Sami Jaffrey paper is that the tornado circular RNA builds up to ridiculously high levels in the cell. So now we have some RNA that we can maybe stick a barcode on and capture. So this is what JB and Sam in the Chanduri lab did. But of course, they were much more interested in capturing it in single-cell RNA sequencing. So when they did this, and they're interested in doing it at higher MOI, they used this piggyback integration and integrated in a plasmid that had both the circular RNA, but then also other promoters. And when you do this, you just get a huge increase in the number of these barcodes that are detected per cell, and an incredible increase in the amount of capture you're getting per cell. And so this barcode capture issue is actually a big issue in the single-cell RNA-seq, just because single-cell RNA-seq is so expensive, and that you really want to make sure that if you're doing like a perturbational screen, every cell is mapped to some perturbation. You don't want to spend half your cells and half your sequencing budget and half your money on cells that don't get mapped to a perturbation, just like we didn't for FISSEQ. So when we apply this and just have a barcode that's expressed as a circular RNA, independent, now decoupling that from the expression of the transgene itself that we want to express, we get way more dots per cell. So you can see this here. And so you get a number of dots per cell that correspond to the original guide RNA paper, and potentially even higher. So this is one way we kind of really improve the technology, I think. Although there's other ways I think that we could have proceeded. This way worked really well. So when we generate this big data set, we need to come up with a way to analyze it. And so working with Nick Bradley, who's a UW-CS undergrad student, to develop a pipeline to analyze something like 3 terabytes of data we get for every replicate of a library. So we got to stitch and align every well. So every well we want to stitch into a large well image that's up to like 100,000 by 100,000 pixels in size. And then we align all of the 12 bases together And we do the stitching and alignment all in one step. I think this is kind of what led to problems before, where it's actually difficult to stitch and not have these micro artifacts that mess up the alignment. But if you stitch and align in one step, then you can generate well-level aligned images for every base that are registered, so that every dot you can kind of look at in every base and see how the color changes and read off the barcode sequence. Then we do dot detection on those stitched and aligned images, and we also do cell segmentation to assign every pixel to a particular cell. And then from those dot detection, we do base calling and reassembly. And so from that, basically, from cells, once we collapse the cells, we can get a cell ID. So for every cell in the experiment, of which there's many millions, a read that corresponds to some 12 base pair barcode sequence, and then a number of counts that that cell has. Some cells may have multiple barcodes, as I'll talk about in a sec. So when we did this, we got when we actually applied this pipeline, not to our data, but to data that was published before in the Feldman and Singh paper, we found that we got a near identical result to their pipeline, but we were also able to get the stitching to work, which we really liked, which I'll talk about in a second, why this is advantageous. And so in recent experiments we've done on a small piece of Lamin A, we've been able to get more than 6 million cells segmented and more than 4 million assigned to a barcode sequence. And so this is kind of, we're able to start scaling the technology up to the point where we can do thousands or even tens of thousands of variants in this library. Okay, so with that, we're kind of analyzing the data right now. So I'm going to skip over exactly what the data looks like, but I'll talk about some of the other next steps that we're doing to try to get this to even higher scale and potentially to work in different cell types. And so one of the next steps is that we want to be able to get this to work not just in U2OS cells, which are a osteosarcoma cancer cell line, but really in biologically relevant cell systems like iPSCs and their potentially their derivatives as well. So to do that, the challenge we actually faced was not necessarily doing physics in those cell types, although that's also potentially challenging. But even to get the transgene expressed in those cell types, I think is a little bit challenging because there's so much transgene silencing. So recall that what we were doing initially was using the landing pad, which works really well in cell lines like U2OS and HEC293T. And what the landing pad does is a genomically integrated landing pad, and then you can use a recombinase to take a plasmid that you transfect into the cells and then integrate that in at the landing pad position so that the promoter from the landing pad expresses whatever transgenic cassette is on that plasmid. And so this kind of gets you uniform expression of whatever transgene or transgene library you want in a particular genomic location. Unfortunately, these seem to silence in iPSCs, and especially during iPSC differentiation. And so to really kind of get a system that works well in iPSCs, we wanted to, and potentially to work well in a bunch of different cell types, we wanted to use a different system to express variants. And so we first thought about, we thought immediately about using the piggyback system that the Shinduri lab has been using to do this piggyback incorporation of genes randomly into the genome. And unfortunately with this, you get potentially high MOIs, but we've been working on titrating down the expression of the piggyback system to try to get one variant per cell. And the nice thing about PhysSeq is that it's a single cell assay. And so you can really remove the cells that have 2 or 3 or 5 integrations per cell later on, as long as those don't take away too many of your cells. And so this piggyback expression system has already been demonstrated to work well for expressing things in iPSCs and MESCs, and that's why we kind of went with it. And so we added on a whole bunch of bells and whistles that help with this transgene silencing problem. So we're of course trying to express this GFP tag protein variant and also a circular barcode from a U6 promoter that's tagged, and that barcode is tied to the variant of interest. But also we wanted to express a selection marker, and the puromycin selection works amazingly well in iPSCs. You can really get 100% after pura selection alone. And we also added other sequences that try to insulate what we're trying to integrate into the genome from nearby genomic sequences that might change expression, like the insulator sequences I've shown you. And on top of that, we added this UCOE enhancer sequence, which has been shown to mitigate transgene silencing in iPSCs. And then also, there's this recent paper that showed that the human silencing hub is particularly active, which is particularly active in iPSCs. It actually recognizes intronic sequences and seems to silence specifically intronless genes. So we added some intronic sequences to the 3' UTR that actually we removed some of the splice sites from those intronic sequences to make sure that they don't splice themselves out. And so these intronic sequences were shown in that paper to reduce silencing. The sequences themselves, not even the splicing, which is fascinating. But when you kind of make all these changes and try to get the Lamin A library to express, it actually expresses really well, even with the aggregation phenotype in IPS. So this now you're seeing it in IPSCs. Okay, so something else we wanted to do is look at other proteins. So we wanted to ask, this strategy works well for Lamin A, but it really is an incredibly general method that allows you to do single cell level correlation of phenotypes and genotypes. And so can we now generalize this to a whole bunch of other proteins? So one of the ones we thought about immediately, because our lab has some experience with it, is the PTEN protein, and then also the CFTR protein. So I'll talk about this in a sec, but just to kind of motivate why we're interested in this, pathogenic mislocalization is a really common mechanism of pathogenesis proteome-wide, or at least is believed to be. So there's this recent paper that came out in bioRxiv, and they found that they looked at something like 3,000 variants and over 3,000 variants in 1,000 genes. And they found that a large percentage of genes, up to 10%, had at least one mislocalized variant, even if you just look at only one variant in that gene. And then as you look at more and more variants, the fraction of genes that have at least one mislocalized variant increases. And so one of the claims they made in the paper was that probably something like 1/6 of genes probably have variants that mislocalize. So this mislocalization is like an obvious mechanism for potential pathogenicity. So that's kind of why we think that this technology is general. And so the hope is now we can also try to get it to work on PTEN and other genes. So this was recently published by the Mikko Taipale lab. And so, of course, for PTEN, we've done this, used this VAMP-seq method. So Kenny Matrake in our lab was a postdoc, developed this VAMP-seq method that allows us to score variants based on abundance. And then there's also this yeast scan that allows us to score variants based on activity in yeast. And so the problem is that most PTEN VUS can't be reclassified with the current data set. So Sean in our lab looked at trying to reclassify VUS and PTEN and found that something like only 15% of the variants, and you can see in those variants that are checkered right here, could be reclassified from VUS to likely pathogenic in PTEN. And that's because that there's all these variants in PTEN that seem to have neither loss of abundance or activity, but are potentially pathogenic. And so that seems to and necessitate now a new technology to understand what these PTEN variants are doing. And this is specifically the N-terminus, but really throughout the whole protein, that seems to be variants that mislocalize that cause pathogenicity. So there's a whole bunch of different papers, but here's one I just pulled out where they looked at wild type and this N-terminal variant K13R and found that specifically that the K13R variant gets less PTEN into the nucleus, and that nuclear PTEN, and specifically because it gets less PTEN into the nucleus, it seems to aggregate PTEN's tumor suppressor effect and potentially be oncogenic. And so what we did is we expressed a PTEN variant library in iPSCs. And so once we did this, we immediately saw that, of course, there's these abundance phenotypes. So here, green is PTEN library, and DAPI is the nucleus of iPSCs. So you can see huge differences in the expression level. And that's likely due to the fact that a lot of these PTEN variants that we're looking at are lowering abundance, just like what Kenny found in his scan. But you see also huge differences in the nuclear to cytoplasmic localization of variants. And so we kind of saw this heterogeneous nuclear cytoplasmic localization, and we're pretty happy. So this is kind of what we're going to try to go after next. And the cool thing about putting PTEN into iPSCs is you can also then take those iPSCs and differentiate them into neurons. And so, and those using, say, NGN2, forced NGN2 expression, and PTEN causes neuronal phenotypes. So we're also interested in doing this and potentially trying to seek or other scans of PTEN. Similarly, CFTR is a protein that I shouldn't have to introduce too much. It's the chloride channel whose variants cause cystic fibrosis, a really common genetic disease that causes a huge amount of morbidity and mortality. And so we expressed a library of CFTR variants using our transgenic expression system in iPSC and found that you see, again, a huge heterogeneity in the level of expression that you get, which also makes sense, right? So there's all these variants that in CFTR that either get degraded right when they're made or get degraded in the, some part of the chain between expression and the cell membrane. And so we find that some variants, but not all of them, are making it to the cell surface. And so what's exciting about doing this on CFTR variants, where we can kind of see the cell surface and see which variants make it there, is that there's these drugs, so VX-809 or lumacaftor, that's been developed that specifically help CFTR variants make it to the cell surface. And so we can not only do something like a FISI scan, but we can also do that in the context of drugs. And so asking which variants specifically are helped along by the drug to make it to the cell surface. Okay, so something else that we're interested in is cellular morphology. So something that I've been talking about this whole time is just localization, but cellular morphology is also something that we're interested in. I'm actually running out of time here, so I'm going to kind of go quickly through this. But basically, we got the N. Carpenter cell painting assay to work really well in cells, so we can stain all these different cellular structures. And so nucleus, the variant of the protein you're interested in, but also things like actin, plasma membrane, mitochondria. And we can then remove all these dyes and then do in situ sequencing. That's also, we think, could be potentially very interesting, even for Lamin A, because we know Lamin A interacts with things like actin. And then lastly, once we get the data, we're interested in, of course, trying to use this to help genetic test interpretation for Lamin A and these other diseases as well. So with that, sorry for running a little bit over time. Just wanted to acknowledge Doug Fowler's lab. I mean, I think he's been an incredible mentor in helping me develop this technology and apply it to a whole bunch of different proteins. Helped me solve problems in cell culture and all these other sort of technical issues that we've been running into. And then a lot of other people that we've been kind of working with and talking to in genome sciences. And then, of course, like both this CMAP and IGVF grants.
Thank you, Sriram, for the nice presentation. I have a few questions for you. Is the mislocalization rates proportional to fitness defect? For proteins that have multiple phenotypes, how can one correlate fitness, aggregation, and mislocalization phenotypes using your method?
That's a good question. I think that for Lamin A, it's going to be pretty challenging to correlate. So Lamin A specifically causes disease in like the 5th or 6th decade. So I think it's going to be pretty challenging to be able to actually make a ton of claims about exactly how it changes, like say reproductive fitness, because a lot of the disease often happens. You often get dilated cardiomyopathy after you reproduce. So I don't know if that's the right protein to go after. In terms of cellular fitness, we couldn't find, by just expressing Lamin A variants, we couldn't find huge differences in cell growth due to those variants and the aggregation status of those variants. And so we decided not to try to do a sort of growth-based scan for that reason. For other proteins, there's certainly fitness effects. So for PTEN, we're hoping that if we can get the library differentiated in neurons, that there will be changes in fitness in neurons of that PTEN variance, just meaning in terms of the growth of the neurons. But it's actually reversed in that the sense that pathogenic variants might actually cause neurons to grow faster because it's a tumor suppressor gene. And then, I don't think I'm enough of an expert on like Cowden syndrome to know about the answer to that question, but I think we're going to try to correlate basically with variants in ClinVar and other databases, and hopefully be able to have something to say about that.
Okay, great. I'm going to read one more question, and you feel free to answer the other questions in the Q&A. Your lab also has the great photo activate and flow technology.
Yeah.
Why did you go with the in situ sec approach versus the flow approach?
That's a great question. So initially we went with the photo activation approach, of course, because that's what our lab published in 2019, 2020. And we found that this actually worked okay for one small region of the protein, but it really didn't seem to generalize well once the phenotypes became sufficiently rare. And so different parts of the protein might have different rates of aggregation. And that might be one reason why we weren't getting highly reproducible data once we started going to other regions of the protein. In addition, we really think that there's phenotypes here other than just aggregation. So like things like the lamina itself kind of breaking, the nuclear membrane itself breaking and rupturing are another phenotype that is more rare. And what's amazing to me is that even though the photoactivate and sort in principle is much easier to do, the FISSEQ actually ended up being getting us more cell throughput at the end. And so kind of these methods like in situ sequencing, I think have now come to the point where yes, I think they're a lot of work, but they're really becoming quite generalizable and applicable and doable by many labs. In addition, a third reason is that David Feldman, who was one of the co-authors in that Cell paper, came to David Baker's lab in UW and built this microscope for FISSEQ. And so now access to that microscope, in addition to a whole bunch of other things, kind of moved us in this direction. And I think comparing the two technologies, really what VCS is very, very good for is it's very good at sorting out cells that then you can then do complicated measurements on. So not just DNA sequencing, but things like single cell sequencing, which I think is really where it shines. And this is the photoactivate approach. Whereas where I think FISSEQ shines is for doing these variant scans.
Okay, great. Thank you so much.